So hi everyone, my name is Elaine, and today I will be talking to you all today about climate niche variation of Hawaiian Psychotria across an endemic island diversification. So to start off with, oftentimes in ecological research we come across this big question, more oftentimes generalized, asking why are some species more common and why are some species considered more rare? And in our case, we want to look at species distribution as a way to understand commonness and rarity. And in order to measure distribution, we want to look at a geographic range size that a species can occupy. And so, for example, you could see that one species can occupy a really large and broad range size, and on the other hand, a species can be very limited and occupy a very much restricted range size, as shown in orange, whereas the others are in blue or green. And as you may wonder, why is this the case? And so we've come across three hypotheses that try to get at this question, one being species age. So are rare species simply just young or old species? The second being dispersal. So does dispersal play a part in whether better dispersers will ultimately result in a wider dispersal across a larger range? And there are traits such as fruit and height that can get at understanding dispersibility. And the third hypothesis is species ecological niche characteristics, which essentially can a species' certain set of conditions affect where they inhabit or possibly expand their habitats. And in our case, we want to look at niche characteristics evolving climate. And so far, our interested study system is Psychotria. So Psychotria is one of the most species-rich genera of flowering plants in the Rubiaceae family with around 1,600 species. They're found worldwide and they vary greatly in their geographic range sizes. And more importantly, our main focus of this talk is on the 11 species of Hawaiian Psychotria. And they are endemic to the Hawaiian Islands, and they are found across the different 6 islands. And what they exhibit is this older to younger colonization example, which I'll go into next slide. So what I have here is just a general map of the Hawaiian Islands, on Kauai being the top left right-hand most, oldest island, around 5 million years. And as you go down towards the right end here of the big island of Hawaii, it's around less than 500,000 years. And what's really interesting is that, as you can see from the miniature version map there, the Molokai, Lanai, and Maui were once connected as a collective island known as Maui Nui. And here is just another diagram of the same, but showing the distribution of these 11 species. And 4 of which I want to point out are Hauptia and grandiflora, which are on the left top. And they are considered 2 of the rarest US native species. And they are only found on Kauai. But as you can see, when we go towards the direction of the younger islands, like Hawaiiensis down here, they occupy up to 3 islands. And you may wonder, why are we seeing such a difference since these 11 species are such closely related and shared in their species relationships? So the first objective is to look at their relationships in terms of their phylogeny and how looking at their evolutionary history can show us how they are closely related. And in order to do so, we've incorporated both nuclear and chloroplasts genetic data, and the chloroplasts will help us trace the history of seed dispersal as well. And last, we want to infer the climatic niches of Hawaiian Psychotria using georeference records, so longitudinal and latitudinal data where they are currently found. And in order to build the phylogeny, we just simply extracted samples that we have obtained from the botanical garden in Kauai. We underwent PCR amplification and ultimately used these edited sequences in Bayesian phylogenetic inference methods in this program called BEAST. And in terms of modeling the niche of climatic niches, we've input these bioclimatic variables that are extracted from BioClim, and these variables range from mean temperature all the way to precipitation. As you can see, they vary across the islands greatly. And we also input these latitudinal and longitudinal data records of where these species are currently found. And they're represented by those black dots. And ultimately what happens in Maxent is that they will produce these figures here known as heat maps. And these heat maps are essentially showing a suitable habitat, which in this case is our metric of predicted climatic niche. And so the greener areas show a more suitable habitat, and the pink to white areas are less suitable. And for the results, we were able to successfully incorporate the both nuclear and chloroplast genes. And as you can see by the arrow indicated and by the color coding, that the all color coded ones are the Hawaiian species. And the older species are able to group together monophyletically. But as you could move on the top of the phylogeny towards the more recently diverse species, also known as the younger species, you could find that Mauiensis, for example, in brown, are located in two different clades. So it suggests that maybe due to the lack of time to diverge into specific species is occurring right now. And also, since they all share a common single ancestor, we were able to infer that there was a single colonization event of these Hawaiian Psychotria species onto the islands. So in terms of building the ecological niche models, we produced heat maps for 11 species. 3 I have here, for example, showing just in general that the climatic niches that were predicted vary greatly. For grandiflora, being on the far leftmost, is very restricted. The potential range is only shown on Kauai, but as we move on towards Mauiensis on the far right here, you can see that its suitable habitat is very much broader, and we predict a larger potential range because of that. And what I have here is just a condensed version of the phylogeny I showed earlier, right next to all the suitable habitat heat maps. And it's in general showing the more basal, older lineages down here are more restricted in its predicted climatic niche. But as we move on up the phylogeny, you can see that it is much more broader for the younger species. And so with these predicted climatic niche, we wanted to be able to quantify it, so we measured its niche breadth. And the figure here shows on the x-axis, which is a no distance from root, which we use as a metric for age. And as we move on towards the right of the x-axis, those are the younger species, and you can see that its niche breadth is much greater than the older species. And so a slight transition from the climate niche, we also looked at the dispersal traits, and we were able to compile data looking at its fruit size and the height of these 11 species. And we found that in general, as you see, the fruit size don't vary much across these species, but in fact, height actually showed a significant difference. Some, like grandiflora, you can see it goes up to like 5 meters or so with Greenwellii, but as we go down towards Meridiana, for instance, it can reach up to 25 meters. So this difference in height is pretty alarming. And from there, we did the preliminary initial multiple regression tests to tested the species age traits from the divergent times of the phylogeny, incorporated with niche breadth, and also the height and fruit size to see which factors play a greater part in whether we could see the number of island occupancy is greatly affected. And the results show that height plays the most significant factor. And to wrap this all up, just looking at the phylogeny, we were able to incorporate both nuclear and chloroplast data, and it largely supports the previous work. And we could see from the niche models that the oldest diverging Hawaiian species show narrower niches than younger species, but the significance of plant height is predictive of the island occupied, and it goes to show that Moranion heifers, which is also greatly distributed across 5 of the 6 islands, is shown to be the greatest of the heights as well. And so future directions include adding more samples and continuing with integrating both the niche models and the phylogeny, and also other dispersal traits to get at answering this question. And with that, I'd like to acknowledge all my community members and staff at UCSF and all our collaborators at the Botanical Garden in Kauai, and also the lab members within my lab that helped with the research. So, and also thank you guys for coming.
Mm-hmm.